in which we start a task. So we should select the guidance or the leader of the program. That purpose, I very call upon Dr. Abdul Aziz Hamid from the Department of Chemistry to use the HOD of the Department of Chemistry Service. Yes, sir. Myself, Dr. Abdul Hamid. I take this honor to announce the nomination of the President of today's international conference. And I very humbly take the name of the head of this institution, Honorable Dr. Sarma Abdul Sattar Ma'am, as the President of today's function. On behalf of all, I second the same and I seek the kind permission to take this program further. Thank you, madam. Thank you. No, sir, please. Thank you, President. Now, for the aims and objectives of this international conference, which is itself a very qualitative conference, as far as its title, its relevance to the present academics. So, I call upon the organizers, one of the organizers of this conference, HOD of Zoology Department, and he is none other than Dr. Mohammed Ismail Mohammed Budai, for the aims and objectives of this conference. Please. Collaborations, professional and research technicians are professional from diverse fields. The aim of the conference are provide global platform for researchers and technicians to share their findings and advancement in scientific and educational research. To encourage interdisciplinary collaborations by bringing together experts from various domains, facilitating the perspectives and innovative solutions. To promote research and development by addressing contemporary challenges in science, technology, and education through academic discussion and practical insights. To facilitate networking and knowledge sharing among research scholars, industry professionals, and students, promoting academic and professional growth. The objectives of the conference are to organize keynote sessions, panel discussions, and technical presentations featuring eminent scientists, educators, and industry leaders. To encourage young researchers and students to actively participate, present their work, and gain constructive feedback from experts, and to explore innovative teaching methodologies and technological interventions that can enhance educational system and research efforts. Through this conference, we aspire to create an environment where ideas flourish, where collaborations and strengthen, and meaningful contributions are made to the scientific and academic community. Let us work together to make this conference a remarkable and successful event, wishing you all an engaging and fruitful experience. Thank you. He is one of the organizers along with Dr. J.A.T. And both the departments in collaboration are organizing this conference. And now it's time for the welcoming speech of all the attendees, all the speakers and participants. I know that Dr. I very respectfully call upon Principal Dr. Salma Mustafa, and she is also the president of this conference. Please.
Hello. Good morning to all of you. It is my great pleasure and honor to welcome you all in this esteemed international multidisciplinary conference on current scientific and educational research organized by Department of Botany and Zoology. On behalf of J.A.T Governing Council, especially Honorable Secretary Alhaj Nehal Ahmad Ansari Sahib, Honorable Chairman Ansari Anis Ahmad Sahib, Honorable Treasurer Advocate Niaz Ahmad Lodhi Sahib, Principal, Teaching and Non-Teaching Staff and students, I extend a warm welcome, a greeting to our distinguished keynote speaker, esteemed researchers, academicians, industry experts, and participants from across the globe. This conference serves as a dynamic platform for the exchange of innovative ideas, groundbreaking research, and interdisciplinary collaborations. As we navigate the ever-evolving landscape of science and education, it is imperative that we come together to discuss, explore, and address the challenges and opportunities that shape our fields. The theme and topics covers in this conference highlight the critical role of research in advancing knowledge and fostering solution to real world problems. Through our valuable contribution, we aim to inspire new perspectives, strengthen academic networks, and pave the way for future developments in various disciplines. It gives me immense pleasure to announce here that for this international conference, we received total 133 participants, out of which for paper presentation, we got 100 registration, and for paper publication, 36 papers are selected for publication in International Journal of Indian Association of Productivity, Quality and Reliability. I congratulate to all the organizing committee members, especially Dr. Ismail, Dr. Yahya Khan, Dr. Adil, Professor Munawwar, Professor Naveed Akhtar, Professor Naeem, Mr. Abdul Hasib. I encourage you all to actively engage in the discussions, share your insights, and make the most of this opportunity to collaborate with like-minded professionals. Let this conference be a catalyst for new ideas and meaningful partnership that contribute to the progress of science and education globally. Once again, I extend my heartfelt gratitude to all the organizers, sponsors, speakers, and participants for making this event possible. I wish you all a productive and enriching conference experience. I would like to congratulate and give the congratulation for the organizing this international event, make it a reality, and happy Science Day. And it's honor for us to celebrate Science Day in the form of international conference. And for this selection of the day, credit goes to all the team members. They decided to celebrate this day as Science Day as international conference organization event. Then I congratulate to all the members. Thank you. Thank you very much. Have a nice and wonderful day. Thank you.
For this brief and accurate welcome speech, and I have mentioned in the speech that how valuable this conference is going to be, and it's a title, is really the need of the time. And one more thing I would like to share with all the participants, that man has guided us, led us in all the pictures that the college has been undertaking since the last 2 years, especially after completing our first cycle of man, in which we earned good plus. Now, the time has come to release the tools of communication issues of this conference. Dear participants, it's a very heartening news for all of us that this conference, 36 papers have been included, and all are very much relevant, very much qualitative in its theme, in their themes, as well as the subject matter. So now I respectfully call upon all the key contributors to release the publication of the issues. And on top page, especially. It's,
To all the team. It's honor for me to be a part of this release of cover page of International Journal. I congratulate all the contributors, also all the participants and paper presenters. Congratulations. Thank you.
We conclude generally our inauguration session. For thanks and for this purpose, I really humbly call upon Dr. J.A.T. for the board of finance.
Thank you, sir. First of all, I would like to thank our honorable management of J.A.T institutions there for continuous inspiration and giving us permission to organize the international conference on the occasion of National Science Day. Then I would like to thank our honorable principal ma'am. Every time she had guided us and encouraging us for doing such practices in the field of research, in the field of education, and in the field of academic. Then I would like to thank our resource person that they have joined right on the time, Dr. Reggie sir. Thank you for your acceptance. Thank you. And thank you all the participants and my team members from J.A.T institutions for successful organizing of this workshop or this international conference. Thank you all of you.
Technical session.
So now it's time to start formal technical session 1. In that, for that technical session, we have a very wonderful resource person, none other than Dr. Reggie Joseph Burgess sir. He is working as an assistant professor at Madras Christian College, affiliated to Chennai University, Madras University. Sir is having PhD in botany in 2013 from Tropical Botanical Garden and Research Institute under University of Kerala in 2013, entitled In Vitro Studies and Genetic Diversity Analysis in some selected bamboo species. On his credit, 2 Indian design patents, and there are list of international research paper in elsewhere Scopus, and all that. So with a huge academic and research background, now I welcome Dr. Reggie sir to please deliver a first technical session. Welcome, sir.
Thank you, sir. Thank you, sir. Sir, am I audible? Sir, am I audible? Yes.
Sir, audio is not working here.
Sir, am I audible? Am I audible now, sir? Hello. Sir, am I audible? I
Yes,
can
sir,
hear
yes, sir.
you.
Now you are audible. Sir.
Sir, am I audible?
Yes, sir, you are audible, sir.
Okay, thank you. Thank you. Thank you very much, and Dr. Yahya Khan, and thank you very much for inviting me for as a resource person for this conference. And I congratulate the college authorities and you especially for taking up this conference into a big level as a part of a small like a Science Day celebration program. So, shall I share my PPT? Yeah, is my PPT visible? Is my PPT visible? Hello, sir.
Yes, sir, it's visible.
Yes, thank you. Thank you. So today, yeah, as an interdisciplinary conference, I would like to share a few thoughts on mining of genes on prospective value from plants, right? So first, one second. Yeah, first of all, we should see what is gene mining, and many of you may be well aware about, or many of you might have heard about gene mining, and many will be having a doubt why we have to go for it. Different difference of opinions may be there, but okay, we will see at the end of the session. You may be knowing like what is its importance and why we have to do. So first, with a few slides on introduction, I will come into our own experience, our own work, which we have done in our lab. So that also will be shared. So gene mining is nothing. It is the identification and isolation of genes, which may be very much beneficial, which may be having very much beneficial traits from the plants. So we are focusing mainly on plants, and also gene mining, it is not pertaining to plants alone. It is applicable to the animals and microbes even. So it has got a tremendous application in the field of medicine, like many genes which are responsible for secondary metabolite production, leading to pharmaceutical advancement. Because you may be knowing that more than 50 percentage of the allopathic single molecule drug are derived from plant sources, like the Taxols, the paclitaxel, that is anti-cancer drug, or artemisinin, which has got a very high anti-malarial potential. And then morphine, which may be misused also, but it may be very much helpful in the pain management when the surgical sessions are done, and so on. There are many other molecules also. So if you are finding out the genes which are mainly responsible for the production of this secondary metabolite, that I will be talking at the end, so which will be of very much useful. Then and it has got an application in agriculture also, like identifying the genes will be helping, which will be which will be having, which will be responsible for the stress tolerance or pest resistance or crop yield improvement, definitely can help in agriculture. So, as what the when we are facing the global challenges, the food scarcity as a global challenge, definitely biotechnology can play a role in producing more of food, right? So that can be done because there is not much of land which we can take from from the earth for cultivation. The only possible way is to cultivate the improvement in the agriculture, so improvement in the productivity by modern agricultural practices. And we can have the pest resistant genes. So we can, by which we can make pest resistant crops that can be from, like now it is mainly pertaining to the genes which have been taken from the Bacillus thuringiensis. That is, you might have heard about the BT cotton or BT maize. But there are many other candidate genes from even plants. So whenever we are talking about like the genes which have been taken from the bacteria, people will be thinking like, okay, it's a transgenic crop which will be having so much of other problem. But we have several other gene candidates which can be taken even from the plant as such. So that also we can mine, we can find out what are the genes which, like the protease inhibitors, or what the chitinase gene, which will be helping us to make the plant resistant towards the pest. Then there are many drought tolerant genes like DREB or Lea proteins, which will be there in many of this climate resilient crops like the millets. Millets, especially you may be knowing that they can be grown in drought conditions. So there are many genes which will be giving those particular characteristics to those plants. So those genes also can be like find out, and if you are incorporating them into the plants like the rice or other uh, varieties or species of plants which require more of water. So we can even make, which will be like very much helpful in agriculture. Then NPR1 gene in Arabidopsis, which will be enhancing the disease resistance in crop. So that is also a well-known gene which has been already isolated. Then salt-tolerant genes for the improved productivity in saline soils that you might have recently heard about. Like even China is having some rice varieties which even you can grow on seashores. So if we can think of that kind of a thing, definitely we are having a lot of the seashores. So if we start cultivating them, if we are cultivating the rice varieties, even the seashore, we can make use of that also. Then apart from that, in like industry, we have many plant-derived enzymes are being used, or biofuels are being used, or many other sustainable materials like the bioplastics and so on. So which also are produced by the genes by different plants, genes which is present in different plants. So that also can be mined. We can even produce in larger scales for the use of for the humanitarian use. So the thing is that we have cellulase genes, the plant and microbial sources are there for the biofuel production from lignocellulosic biomass, or the rubber synthesis genes. You may be knowing that the natural rubber which we are getting from the Hevea brasiliensis. And if you are identifying, and if we can clone those genes into even microbes, we may be able to produce natural rubber without even the plantations, right? So that also is possible. Then bioplastic production, but like the PHA polyhydroxyalkanate or polyhydroxybutyrate synthesis, uh, also the genes which can be incorporated, we can identify and we can incorporate for the sustainable alternatives to petroleum-based plastics, which will be a menace. The normal petroleum-based plastic, you may be knowing, we cannot think of plastic, but how we can utilize in a better way. That is what we have to think. And the other alternative is like the bioplastics, which are degradable. So that also is there. Then essential oil synthesis, like which will be having much of application in cosmetics, perfumes, and flavors. So that these are the major. Then we can go for these are some of the applications which will be there in what the gene mining. Then there are several strategies which we can use for gene mining in plants, and everyone knows about the traditional methods like classical breeding and trait selection, which we will be having. Okay, some plants which are having some specific traits that will be isolated, that can be taken, and we can isolate those things, and we can put that genes by selective breeding, and we can pass on to the other plants if they are compatible enough for hybridization. But by modern biotechnological approaches are also there. Like we can use a comparative genomics, or you can have transcriptomics and RNA sequencing we can use, or functional genomics we can use, or metagenomics and synthetic biology. We can synthetic biological approaches we can use for gene mining. So we will have a glimpse on these things like genomic approaches for gene mining. So we have whole genome sequencing techniques for identifying the gene sequences linked to the beneficial traits. So now the onset of new generation sequencing techniques are available, then we can the sequencing of a whole genome as such is not very difficult. So we can have the genome of the entire genome sequenced of a plant which has got so much beneficial applications. Then we can identify the genes which are beneficial for us. And genome-wide association studies like identifying genomic variations responsible for the desired traits like SNPs and all, that we can find out the genome-wide association studies is another way of doing it. And comparative genomics, definitely identifying the conserved and novel genes across the plant species also will be helping in the gene mining. Then transcript techniques, we have RNA sequencing, like identifying differentially expressed genes under specific conditions. So that is what I'm going to talk more on it. Then we can have expressed sequence stacks, like short sequences that represent the actively transcribing genes. Then microarrays also is available for the high throughput analysis gene expressions. Then functional genomics like the gene knockout and overexpression studies also can be done for understanding the gene function in secondary metabolic pathways. Because I told you I will be coming into the secondary metabolite protection by using gene mining. And CRISPR-Cas9 and RNA approach also is there for validating the role of the candidate genes which we are isolating. And transgenic and mutant screening definitely will help in identifying the beneficial genes for crop improvement. So, and then metagenomics approach also is there. Like we can have studying plant-associated microbiome for novel gene discovery. Like we can identify the novel enzymes for biomass degradation or specific degradation of some compounds and so on. And discovery of bioactive compounds with the pharmaceutical properties also will be possible by a metagenomics approach. Then, mining of genes for metabolite production, like a pathway reconstruction of secondary metabolite synthesis can be done. Say, for example, engineering the hypericum species for increased production of hypericin production. Hypericin is an naphthodianthrone compound, which normally will be there in many of the hypericum species, which is a low volume, high value compound, and it is having mild to moderate antidepressant. It can be used, and it can be used like the pathway can be reconstructed for enhanced reproduction if you are identifying the genes which will be synthesizing this hypericin. And people were trying for long years for the identification of the pathways as such. And we will come to it later. And synthetic biology and heterologous expression expressing the plant genes in microbes or plant-based bioreactors, which can be used like when you are having synthetic biology or heterologous expression. So we are coming to a case study here, what we have done. Here we were working with a plant called Hypericum hookerianum, which is an Indian counterpart of Hypericum perforator. You might have heard about it's a high altitude or temperate species, which is identified as a good source for this hypericin. As I told you, it's a pigment. It's a naphthodionthron pigment, and which is having a high potential for treating mild to moderate depression. And what we found is that we could establish in vitro shoot cultures in this particular species, which is available in our Western Ghats, and it is an endemic species of Western Ghats. And we could control the production. We could find out how we can control. We have got an absolute control on the production of this particular hyperizin in the in vitro conditions by adding auxins exogenously and also by controlling certain what the external parameters like the physical parameters. And what we did was what we achieved is like we could produce two systems. One shoot culture system where it will be like producing very less or nil amount of hyperizin, and another one is many-fold increased production of hyperizin. And what we did was we have isolated the mRNA from both the culture systems, both the systems, like we have isolated the mRNA pool using the standard protocol. Now the kits are available where we can use it for, and we have constructed the cDNA library also by RT-PCR. And we have done a suppression subtractive hybridization with both the pool of cDNA library as like for identifying. This is a specific screening test for finding out the differentially expressed genes which will be there. You may be knowing that the control or the system which will be having very less amount of this hyperizin production will be having less number of or nil mRNAs, which will be responsible for the production of this hyperizin. And the highly producing or the system which will be having high production of hyperizin will be having more of mRNAs. And we have isolated both the total pool of mRNAs, and we were making the cDNA library out with by using RT-PCR, and we were hybridizing them, we were normalizing, and finally we ended up with a few what expressed sequence tags which are pertaining to or which are differentially expressed in the system where this hyperizin was synthesizing. And also what we did was we have taken the cDNAs of the expressed systems, and we have done PCR using degenerate primers, which have been already, because initially even everyone was thinking like this particular compound was getting synthesized in terpenoid pathways, and but we could find out from our lab, we could find out that it is produced in a polyketide synthase pathway. It was not in the terpenoid pathway, because we have got specific blockers for blocking terpenoid pathway, and even after blocking the terpenoid pathway, it was getting synthesized, and we found out that certain polyketide genes are also getting overexpressed when these things are getting synthesized. The hyperizin is getting synthesized, and so we were looking for the possible what the polyketide synthase genes from many plants we have taken, and the sequence have been taken, and we designed degenerate primers for seeing whether these genes are getting expressed in our gland. So that also we have found. And we have got amplified a few of this, the degenerate out of 16 degenerate primers. We found that 5 of them were giving positive results and they were sequenced. And finally, we have gone for a complete isolation of that particular CDNA. Complete length of the CDNA have been isolated by using a 5' RLM race and 3' race. So we could find out like this is the result of the RLM race for 3' and 5', for which we could get like we have got 3 products, PKS1, PKS2, and PKS3. So that was the result. And we got 3 products and we have cloned them and we have gone for the sequence by TA clone. After TA cloning, we have gone for sequencing, and this sequencing of this genes, which revealed the cDNA revealed that polyketide synthase, that is PKS1 gene, is having 1173 base pairs, and it has been already submitted in GenBank also. And another novel gene like PKS2, which was different from the already described genes of chalcone synthase family, having 1179 base pairs. And it has we could found that it is having an altered active site also. And yet another gene, PKS3, having 1171 base pairs with novel subtractions or alterations at catalytic triads and active sites residues also. And both PKS2 and PKS3 were novel forms. So we have done the multiple sequence alignment also to find out all these things. And for PKS1 and PKS2 and PKS3, it is showing very clear what the amino acid replacements in the PKS sequence. So the main difference between the PKS1 and PKS2, so what we found is that in conserved amino acid residues at the active sites, so definitely the the tyrosine at 197 and serine at 338 position in PKS1 are replaced by 1100 and sorry, the 197th position, it was replaced by an isoleucine and glycine at 338 position in PKS2. While PKS3, there is an almost total replacement we have seen from 185 to 202, which includes a strategic replacement at positions at 197, where the tyrosine was replaced by leucine. And similarly, the conserved residues at S339, that is serine 339, is replaced by glycine in PKS3. And these replacements are indicative of possible changes in the substrate specificity of this enzyme. And we found that also this shows that these genes were overexpressed in the system, and we have identified and we are analyzing its activities by using by cloning it, subcloning, and doing it and seeing where exactly it is fitting into the pathway, and trying to overexpress these genes for the production of this hypericin in the in vitro conditions. So these are the salient findings of our work. The reverse transcriptase PCR approach using degenerate primers specific to the conserved domain of PKS from other plant sources coupled with race analysis result in the isolation of 3 full-length PKS cDNAs. And the sequence analysis also showed that the PKS2 and 3 with the characteristic amino acid replacement at active site residues are novel forms. So these two genes actually are novel. And so now the hypericin producing genotypes are identified in our lab, and a process also is optimized for hypericin production through in vitro means, and we have already patented that thing also. So now even we have identified some genes which are involved in this hypericin synthesis also is being identified. And now we are cloning it, and probably we may You may ask like, where is the product? Where is the antidepressant capsule made by this biotechnology? Definitely, I can tell you that it will come in future. So we are working on it, and definitely it may come in future by using a completely biotechnologically produced product, a compound which will be completely produced by genetic engineering will be done at the earliest. So with this, I will just conclude, and thank you very much once again to the organizers. And if any questions are there, I'm ready to clarify your doubts. Thank you.
Thank you, sir. So any of the participant that is having the queries and questions to sir, so please ask. So, sir, as a started with the strategies for the gene mining in plants, and then metagenomic assays, and then my mining genes for secondary metabolic production. So he was a wonderful case study with the hypericum species with the suppression substrate to hybridization, and then amplification of cDNA using degenerate pyromone. So with the isolation of full length cDNA by rapid amplification. So cloning is there, and sequencing of rest products. So sir has having a wonderful work that they had presented. He had presented. So any of the participant? Okay. So thank you, sir. Thank you, Rishi sir, for
Thank you,
a
sir.
wonderful talk. It's been a wonderful talk to you. Now I would like to call Dr. Adil Aziz Momin sir. He is a head of Department of Chemistry from
Thank you.
our college. So I will hand over the session to Dr. Adil sir.
Thank you, sir.
Adil sir, करो ना बस. हाँ, आ जाओ. बैठो. Due to the technical issue, we are unable to contact. Sir.
Hello, very good morning to all of you. I'm Dr. Adil Momin to present vote of thanks, and for that, I very I express my heartfelt gratitude towards Honorable Dr. Regis Joseph, sir. Sir, thank you for accepting our invitation and delivering a wonderful lecture.
Thank you, sir. Thank you.
Topic was so applicative, and it will be really helpful for the young researchers, especially on the occasion of this National Science Day, which has a theme of empowering Indian youth for global leadership in science and innovation under Vikasit Bharat campaign. So, thank you very much, sir, for delivering such a wonderful lecture and sharing your expertise with us. Thank you very much, sir. Finally, I thank all the participants who have joined us in online mode. Thank you for careful listening. Thank you, all of you.
So thank you, sir. Sir, thank you. It was a wonderful to nice to see you here.
Yes, sir. Yes, sir.
A long
Yes,
time.
thank you.
And, so it's one request to you. We have
Yes.
some presentation. So if you have time, you can judge them or see them the presentation. Because one more, we have a keynote speaker
Yes,
from
yes,
Ireland.
yes. Sure,
Ireland.
sure.
So till he will join within a half an hour. So we will utilize that time with the presentation.
Okay, sure, sir. Sure.
Okay. Okay. Thank you, sir. Thank you. So anybody from the participant? She's joined. I think so. Javle Ratnamala ma'am. Javle Ratnamala ma'am. Alpa Patel ma'am. Alpa Patel ma'am. Please unmute yourself.
Yes, hello, sir.
Ratna ma'am, you are co-host to share your presentation.
Sir, actually, they were already, it was informed to me to share the presentation around 12:30, and I'm busy in my college event. So, sir, right now I'm unable. Definitely at
Yeah.
12:30, I will be there for the presentation.
Yeah.
Oh, okay, sir. Okay.
Hello, sir.
Thank you.
Hi, ma'am.
May I audible, sir?
Yes, you are audible.
Dr. Ratnamala Javde. Shall I share my presentation?
Yes, sir. Yes, ma'am. Yeah.
Sir, it is visible.
Yes, ma'am.
Good morning to all of you. Myself, Dr. Ratnamala S. Davde, Assistant Professor, Department of Zoology, Veer Vajekar, ASC College, Funde, at Navi Mumbai. Topic: histopathological study on effect ascorbic acid on lead toxicity in the freshwater bivalves, Laminidae coreneus. Aquatic creatures, especially freshwater bivalves, are seriously threatened by the lead toxicity, which can alter their general physiology and reproductive health. This study assessed the preventive function of ascorbic acid in reducing histopathological effects of lead exposure on the testicular tissue of freshwater bivalves. Our predetermined time periods, specimens were exposed to sublethal level of lead and subset of them also received the ascorbic acid therapy. Histological analysis of the testicular tissue showed that lead-exposed bivalves had significant degenerative alterations such as necrosis, vacuolations, and the cellular atrophy. On the other hand, ascorbic acid-treated bivalves showed less histopathological changes, indicating that it may have preventive antioxidant properties. This result emphasizes the harm that lead poisoning causes the aquatic organisms, reproductive system, and the need of the vitamin C. Material methods. The Laminidae coreneus is the experimental animals were collected from the J.A.T dam at the Python. Then it were acclimatized to laboratory condition for the 4 days. After the acclimatization, active Laminidae coreneus were divided into the 3 groups A, B, and the C. The A group of Laminidae coreneus were kept as a control, while B group were exposed to chronic hours of the heavy metal salts lead nitrate, and group C were exposed to chronic concentration of each heavy metals with the 50 milligram per liter ascorbic acid up to the 20 days. After 20 days, exposure of heavy metals, Laminidae coreneus from the group B were divided into 2 subgroups, that is group D and group E. Group D were allowed to self-cure naturally in normal water, and group E were exposed to 50 milligram ascorbic acid up to the 9 days for the recovery periods. The Laminidae coreneus from the A, B, and C groups were dissected after the 10-20 days, while those group of the E and D recovery were dissected after 3 and 9 days. Their testes were removed and processed for the stain for the histopathological impact of the heavy metal salts. Here is the observations. The normal testes, Laminidae coreneus, see in this picture, the testes of the bivalve consist of the testicular follicles which have a densely populated and the spermatogenic stages. Follicles are surrounded by the intratubular connective tissue with some muscles and the collagen fibers. You have also seen this picture, the germinal epithelium of the follicles rest on the thin basement membrane. The testicular follicles are scattered in the fibrous connective tissue through which the support and the nutrients are provided to the growing spermatozoa. Also, there is the connective tissue contains some bundles of the muscles fiber, which when contract causes the releases of the sperms. The germinal epithelial cells present at the periphery of the testicular follicles have a large nucleus. The size of these stages reduce and also the nuclei get constrict due to the loss of the cytoplasma and the nucleoplasma respectively. The fine nuclei of the stages appears densely stained granular structure. The tails of the sperm being a very narrow beyond the limit of the resolving the power of the microscope used are not visible. The testis under the lead intoxications. You have seen in these slides, there is the lesions which are occurs into the germinal epithelium, which are also damaged, and together neocrotic changes is occurs here. Also, the basement membrane and the intrafollicular connective tissue are affected, and the delaminations of the germinal epithelium and its necrosis is clearly visible. Also, you have seen in this two slides, the recovery for the 3 days recovery and the 9 days recovery. The recovery after the 20 days exposure to the lead in the histological structures in 50 milligram ascorbic acid per liter dechlorinated water and 3 days in the 9 days. You have seen in this recovery slides for the 3 days, there is the germinal epithelium layer is also dense in size, and you have clearly show the result in the 9 days recovery. There is the densely germinal epithelium is present in the testis folliculars. With the help of the, it is the effect of the ascorbic acid. Laid with ascorbic acid after the 20 days, this is the slides. As compared to the total study of this lead, the plain treated lead animals show the majorly damages into the germinal epithelium as compared to the animals which are treated with the lead with the ascorbic acids. You have to see in this slide, the cellular identity is lost at most of the places, but shows the effect of the ascorbic acid in the simultaneous exposure to the lead. The damage induced in the structure of the testicular following tissues structure is the maintained to somewhat the normal levels. At last, the differential staining of the testicular follicle indicates the restorations and the functioning of the testes. Discussions? Ascorbic acid is essential for the normal and development, and in most cases, it is produced endogenously, thus resembling a hormone, plays a major role in tissue synthesis and growth process, and obviously mediates rapid tissue repair in the trauma or the disease conditions. Also, ascorbic acid is thought an excellent reducing agent, able to serve as a donor antioxidant in free radical mediated oxidation process, able to reduce the metals such as all metals for the lead, zinc, ferrous, and the arsenic. Thus, the effect of the other toxic metal can be protected by the ascorbic acid, protected specific binding sites of the receptors by virtue of its ability to reduce the insoluble of the lead, and the ascorbic acid readily forms the salts of the several metals, reduce the activity. Conclusion, in the protections provided by the ascorbic acid against the lead in the present study may be due to its antioxidant activity that has scavenged the ROS generated due to the heavy metals, its role in the collagen synthesis, which has rapidly replaced the basement membrane, and due to its binding metal ions. This is the bibliography. Dr. Some Anusia Butternut Chatterjee, Eliar, Ganta, Faria Sarita, and also the Major Sultana, which are studied on these the heavy metals and the ascorbic effects. Thank you.
Thank you, madam, for a wonderful presentation. So, Reggie sir, can you make?
Yes, sir. Yes, sir. Yes, sir. I'm here. Yes, sir. I'm here.
Ma'am, please on your video, madam.
Video, where is, yes, sir.
Yeah. So, sir, please unmute yourself. Reggie sir.
Yes, sir.
Yes. Yes, sir. Yes, sir.
So,
Yes, madam. Why you have selected this bivalves for this study? Any specific reason for selecting of the material?
Yes, sir. The biology is the bioindicator of the pollution.
Okay. Okay. Okay, you have studied like this antioxidants are reviving its pollution, right? Right? Can you suggest anything for like any application for your work?
Yes, sir.
Yeah.
Sir,
What is?
only I have studied presentation, please. Only I have studied on the ascorbic acid, sir.
Yes. So you are telling that it is reverting back its toxicity, right?
Oh, sir. Yes, sir. Because the vitamin C is the antioxidant.
Yeah, it's a
In many
well-proven. It's a
studies,
well-proven one. It's a well-proven antioxidant. My question is, what is the significance of your study?
if in social, we have studied the first the heavy metals effect. All of you
Okay.
very well known that we are the non-vegetarian. We have eating upon the ocean creatures, for example, the mainly the fish in our food.
Okay.
When we have taking the fish or the aquatic animals, at that time in our meals, we have
Yes.
necessary intake of the ascorbic acids. If they have content their heavy metals in their body, all of you very well known that the heavy metals are not created or not destroyed.
Okay.
So if the fishes or the aquatic animals have a polluted from the any heavy metals, then they restore the heavy metals in their muscles and they
Okay.
go into in our diet and our bodies. So
Okay.
we have bind these heavy metals through the ascorbic acids.
Okay.
We have to need in our meal to taking the ascorbic acids.
Okay. Okay, ma'am. Thank you. Nice presentation.
thank you, madam. And thank you, sir. Thank you, sir, for the question. Any of the participant would you like to question to ma'am? Okay, so please stop your sharing, ma'am.
Yes,
Ashok
yes, sir.
Kadam, now the second presentation we had. Ashok Kadam, sir.
Yes, sir. May I audible?
Yes, sir. Madam, please stop your share.
Yes, sir. Stop. Where is the?
Stop your sharing.
Yes, sir. Yes.
Ashok Kadam, sir.
Yes, sir.
Please share your presentation.
Okay, sir. Sir, my screen is visible. Yes, sir. My screen is visible, sir.
No, 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 sir.
Sir, just 2 minutes. Yes, sir, my screen is visible.
No, sir.
My screen is visible, sir.
No, sir. No, sir.
Okay, sir. Yes, sir. Sir, wait for the 5 minutes. There is some technical problem. That's why we are preparing for that. Yes. Yes, good afternoon, all of you. Here my points is there. Just a few minutes. Let me share the screen. Okay, there is slides of my topic is there. Okay.
Hello, sir.
Yes, sir.
Sir, we have some technical issue. है ना? So just wait for 1 minute, 1 to 2 minutes, sir.
Okay, sir. Due to that technical issue, wait 1 or 2 minutes. We are in progress. Sir, if possible, can you take the next presentation after that?
Hello,
Yes, sir.
sir.
Uh, okay, sir.
Actually, this was by the electricity office. Suddenly, the power is went off. है ना?
Yes,
So
sir.
now it is okay. So share your presentation, sir. It is working or not?
Sir, there is some technical problem, network problem. So, you, if possible, you can take the next presentation. After that, I will give my presentation, sir.
Okay, okay, sir. Okay, okay, okay. No
Okay,
issue.
thank you, sir.
So we have
Thank
next Yogini Pagare, ma'am. अब ये योगिनी पगार योगिनी डॉक्टर प्रेमल रमेश देवरे डॉक्टर देवरे सर
प्रेमल नहीं है, रमेश देवरे।
Is anybody interested for the presentation from the participant? Because two of our participants, they are unable to present because due to their technical issue. And one more thing is that we have, today we have a National Science Day. So most of the participant and the faculty, they are busy in their celebration of the National Science Day. So any of the participant,
Sir, I require only just 5 to 10 minutes because I just finished my work. I require only 10 minutes, sir.
okay, ma'am, no issue, no
Yep.
issue.
Okay, thank you, sir.
Anand Mutha, sir, now you are the co-host. Please on your video, sir. And unmute yourself, sir. नहीं नहीं देखो तो क्या बोल रहा है Mutha sir. I have make you co-host, sir. Already done. Please do your presentation. Yes, sir, it is visible. Yes, sir. Please unmute yourself, sir. Gautam, sir. Please unmute. Anand Mutha, sir. Please, sir.
Hello, Mutha sir. Mutha sir, your PPT is visible, but you are not audible, sir. Can you please unmute yourself? We have made you as a co-host, so you can unmute yourself. Your presentation is visible clearly, but your voice is not clear or totally you are muted one.
Kadam sir.
Okay.
Yes, yes, yes.
Yes, sir. May I audio screen is visible now, sir, right now?
Yes. Yes, sir. Yes, sir.
Sorry to all participants and our all dignitaries members because there is some technical problems in here because I am living in the rural area. There is some network problem. So there is some problems arising. That's why sorry to all the members. Here I am sharing my screen, and in that there is a study of preliminary phytochemical analysis of Plectranthus cuticularis. Okay, one plant is there, and in that my guides from the Sri Muktananda College, Gangapur, Chhatrapati Sambhajinagar. I am the research scholar. Okay.
Sir, please go ahead, sir. Go ahead, sir.
What, sir?
Please go ahead with the presentation, sir.
Yes, here the introductions of my in that nature is that there is a medicinal value which are used in the human life, that is Plectranthus cuticularis is there, and here in that Plectranthus cuticularis, one this plant is members of that family Lamiaceae, and in that this plant mostly perennial plants is there, and also shoots are the annual, but mostly there is a branches, stems of the plant are very branches, and indeed it's cultivated as a medicinal plant in the different countries, and this belonging to that Lamiaceae member. Okay, and semi-succulent plants are there. And in that, this is my laboratory work is that the phytochemical screenings, okay, that is qualitative taste of that plant. And normally a morphology of that plant is given there. Okay, mostly these plants are using the anti-cancerous, antibacterial, anti-diabetic, and antioxidant purposes. And in that investigative study, inside that the plant there is some secondary metabolites are there, like that flavonoids, alkaloids, saponins, glycosides, and tannins is there. Also, it contains a different phytochemical constituents inside that the plant. Here the different species of the plants are given there. That is plant Cooley's plant. Normally, common name of that plant is the Cooley's plant. Sometimes it is grown as a ornamental plant, but it occurs in the forest area as a white plant. Okay, and then here the roots of that plant is the treatment of the worms and alleviate burning of the vegetable oil, and sometimes it is used as a veterinary purposes, and preventing the hair graying, then restoring the gray hair to its normal color. Then mostly we can say it is a medicinal plant, and it contains some aromatic smells because some aromatic compounds are present inside the leaves of that plant, and it contains some oils in that tuber, delicate odors and spicy notes, and essential oils are also present in that, and it shows the antimicrobial activity against some the bacteria and the fungi. Okay, here the material and the methods are given there. I perform the materials and methods. That is first, that is collections of the plant from the wide area. I got these plants in our district, that is Ahilanagar, in particular regions. And after that, collections of that plant, identification next passes I perform at that time. Then prepare the powder form of that leaf and the stems of the plant. Then extract preparations and then perform the different qualitative taste of that plant. Okay. Then here the Plectranthus cuticularis plants are the in this slide. That is it is looking like. Then I use the software extractions in laboratory for extracting some liquid parts or the powdery forms from that plant. Please next slide. Here the extract preparation for the preliminary phase, like that is the Plectranthus cuticularis leaf, then root, and also take the stain and some the solvent like chloroforms or ethanols, and sometimes water as active medium, and make the extract of that plant in incubator for the before that softness extraction. Okay, here the phytochemical test are given there from the alkaloid. There is a particular concentrations are taken and some reagents are added, indicate the particular colors, and then I get the idea that here in that the leaf or the stems of that plant, these phytochemical compounds is present or not. And second, also I take the taste for the tannin, then saponines. Okay, there's are given here, anthocyanins, and then flavonoids. Most common features of these plants is that it contains the flavonoids in that the leaves of that the plant, and in that we take the chemical taste, and I got get it color indicated that present of the flavonoids. Then here steroid taste. Also, I take the amino acid taste, then protein taste, then carbohydrate taste, and phenol taste. Then glycoside and turbinate. This 12 times, and then I got reach up to the particular result that phytochemical taste of lecthanthus tuberati. It shows some medium present and some it show the absent. Here the observation tables are there. In that alkaloid in aqueous extract, it show the positive result, whereas the ethanol it is also positive, but methanol it gets the negative. These are the different solvents that is based on that polarity and non-polarity I have selected. Then chloroform, then petroleum ether, and the acetones is there. And this observation table shows that there are positive indications or the negative indications of that chemical constituent in that plants. Okay. And here I think that we can take the references for this one. Right? To the thank you all of you.
Rajesh sir.
Yes, sir. Yes, sir. Yes, sir. Yeah, it's a very preliminary study, sir. And have you tried for isolating any specific compound from this plant?
Yes, sir. Yes,
Any,
sir. What is the question? Yes, it is preliminary test.
yeah. Have you screened it for some specific compounds? Any specific compounds have you screened? Tested for any specific compound? Because in
No, sir.
Colliers
Yes,
or Plectranthus is
first,
Plectranthus
at
is yes.
time, sir, I take the screening of the preliminary test. That is the qualitative test. And my next
Yeah.
task is that go details in that the quantitative test. For that, I take the GCMS, that is gas and mass spectroscopy or the chromatography technique. Like HPLC is also there, and I am performing that test, but my result is pending is there. But this preliminary test I have performed, and I got the indications of present and absent is there. But after that, the quantitative test, I will get the exact structure of that, the quantity of that chemical concentrate in that plant, and also their exact structures of that chemical compounds present in that.
Okay, sir. Thank you. So we have with our keynote speaker from Ireland, Dr. Shanawaz Jadeja, sir. So, Jadeja, sir, please. Please stop your sharing, sir. Ashok Kadam,
Okay,
sir.
sir. Yes, okay, sir.
Stop your all the sharing.
Okay, sir. Okay.
So, afternoon, everybody. Yeah.
Yes, sir.
Can I share my screen?
Assalamu alaikum.
Walaikum salam. J. Ismail bhai, how are you?
I'm fine. I'm fine. What about you? What is the time over there?
It is 7:15 in the morning.
7 in the morning. Yes, yes.
Sorry, just a
So,
moment.
dear participants,
Yeah.
I'm very happy to announce that our second resource person, Dr. Shanawaz Jadeja, has joined with us. I'm going to introduce a brief introduction of Dr. Shanawaz Jadeja. Dr. Shanawaz is a research scientist at Abbott Rapid Diagnostic, Galway, Ireland. He is a PhD in biochemistry and over 3 years of postdoctoral experience in biochemistry, molecular biology, human genetics, skin biology, and immunology. His current research at the Center for Arthritis and Rheumatoid Diseases at University College Dublin, Ireland. Focuses on immune stromal cell interactions in rheumatoid arthritis. He's completed his PhD in biochemistry from MS University, Baroda in 2020. Before that, he earned an MSc in Forensic Science from Gujarat Forensic Sciences University in 2011. Dr. Jadeja began his professional career as an analyst in biology division at the Goa State Forensic Science Laboratory from 2011 to 2013. Later, he worked as a visiting lecturer at Morarji Desai Institute of Neuropathy and Yogic Science in 2019. Throughout his career, he has published 19 research articles and 3 book chapters in international journals. He has received multiple fellowships, including UGC NET Junior Research Fellowship and several travel grants for international conferences. His research contributions include securing a research grant from Alopecia UK for studying autoimmunity in hair loss. So I request Dr. Shahnawaz to kindly present his research on autoantigen discovery in alopecia. Now, the session is for Dr. Shahnawaz Jadeja. Please, sir.
Yeah, thank you, Dr. Ismail, for kind introduction. And I hope everybody can see my screen.
Yes, yes, you are audible.
Okay, okay. So just to mention, like currently I'm working in Abbott Diagnostics, but in the company policy, we cannot, the research which we are doing there is confidential, so we cannot share the information of that. So I'm presenting my work, which I did in postdoc in UCD, okay, on alopecia, autoantigen discovery in alopecia areata. So yeah, so let's understand what is hair follicle. So hair follicle is a tube-like structure, okay, that surrounds the root and strand of a hair, okay, and it exists in top two layers of the skin, that is dermis and epidermis. Okay. And apart from promoting the hair growth, hair follicles also helps in the wound healing, angiogenesis, and neurogenesis. So these are all the additional advantages of the hair follicles. So why we think that hair follicles are so special? Like usually we don't consider, usually we don't take care of the hair and all those things. Like we don't think much about the hair follicles. Okay, so let's see. Sorry. So hair follicles are the mammal's only trait. So it can be found in only in mammals, not in any other living organisms. And it is a mini organ of multiple cell type. It's kind of it's not a cell, it's a kind of mini organ. Okay, multiple cell types are involved. It includes the blood system, nervous system, immune and muscle system. So all the other systems are also interacting with the hair follicles. And hair follicles are the source and target of hormones. And the hair grows in cycles. Okay, it's a very interesting that hair grows in cycle of different cell division, cell differentiation, cell death at a time. And this is the only adult tissue to do so. So there are multiple types, or one hair follicle can produce multiple different types of hairs. So in uterus, when the baby is in the womb of the mother, in uterus, the type of the hair which is found on the baby is called as lanugo hair. After that, in adults, adults and children, you can see the vellus hair. So vellus hairs are the small hairs that we have, जिसको हम रुई बोलते हैं, or then the terminal hair, which are the proper long hairs that we see. So hair follicles, we can see here, sorry for this. We can see here, like there are several, in the scalp, there are different types of hair follicle, but some are small and some are big. So we can see in the scalp only, there are different kind of hair follicles. hair fibers, and the same hair follicle produces the different type of hair fibers which differ in their caliber, in the diameter, in the length, modulation, color, growth, and all the cycling synchrony. So I'll mention that later. So let's learn about some of the basic facts about hair follicles. So there are around 5 million hairs on our body, and only 2% that we found is on our scalp. And the types are three, as we discussed earlier: lanugo, vellus, and terminal hairs. Okay, and hairs are already formed by 16 weeks of gestation in the womb of the mother. And hair grows around 1 centimeter fiber per month on the scalp, and cutting and shaving has no effect on the hair growth. Okay, and normal scalp shedding is around 100 per day. Scalp hair follicles cycles around 10 to 20 times per lifetime, and different based on the anthropological division of the human nature. Like you can see, there are three different races, three different races: Mongoloid, Caucasoid, and African. So they also differ in the caliber and the diameter of the hair fiber. So Mongoloid, you have circular hair fibers, and Caucasian, the hair fibers are little oval, and in African, the hair fibers are elliptical. So there are different forms of hair exist in based on the different races of the human. So let's understand what are the stages of hair growth. So there are basically three major stages of the hair growth: anagen, which is a growth phase; catagen, which is a transition phase; and telogen, which is a resting phase. So here we can see in anagen, the most of the hair follicles in our body are 18 to 90% are anagen, and they grows around 3 to 5 years. And the catagen life cycle, you will find around 2% of the hair follicles in the catagen phase, and the time period of the catagen will be around 4 weeks or a month or so. And the telogen of the resting phase, or where the hair follicle is not producing any hair fiber. Okay, so this is around 3 to 4 months, and around 8 to 10% of the body hair follicles in our body are in telogen phase at a time. So then again it cycles back to the anagen. Okay, after telogen it again cycles back to anagen. The growth factors are coming into play, and they start growing the hair follicles. So each strand of the hair on the human body is at its own stage of development. So in the scalp, we can see, for example, in the scalp, we can see we have all full grown hair fibers, but there will be different hair follicles at different stage of size. Some will be at anagen, some will be at catagen, some will be at telogen phase. And one cycle is complete, it restarts, and a new strands of the hair begins to form. Let's understand the relationship between hormones and hair growth. So during the puberty, so there is an androgen-driven vellus to terminus hair follicle transformation. So we can see, for example, in beard. Okay, so during the puberty, in the childhood, you can see. So the short and fine unpigmented hairs are there, so vellus hairs, and when the androgen starts producing, you will see that the hair follicles are growing thicker and long, and then we can see that during the age of the puberty. And in the adulthood, like when at the late stage, in the old age, like usually we see balding, and balding we can see that the terminal hairs are becoming more to vellus hairs. Okay, based on the androgens. So let's understand what are the major hair loss disorders. So the mostly we can see androgenetic alopecia. So this is androgenic alopecia is usually found in two patterns, like male pattern and female pattern, and most of the people who are suffering with the hair loss will have androgenetic alopecia. This is So yeah, as we can see here, it can affect around 50 to 40%, 50% in the males and 40% in the female due to the age. And around 85% you will see that there is a positive family history. And there is a definitely role of androgens in the pathogenesis. Alopecia areata. So alopecia areata, there are different types. You can see that there are patchy hair losses. You say it's a striking difference between the androgenetic alopecia and the alopecia areata, that you will see the hair loss in a patchy manner. There is a not a complete followed rule for the hair loss. Like here we can see that you will see the baldness going in a gradually, you are losing your hair in a area in your scalp or in a other part of the body. But in patchy alopecia areata, there is not any strict rule or any followed pattern for that. So you will see that round patches around the beard and the scalp. And another type of the alopecia areata is totalis, and totalis means that the complete hair loss on the scalp. And alopecia universalis, it means that there is a no hair no hair growth at any part of the body throughout the body. Okay, so it affects around 2% of the population, and around 30% family history is there. 75% of cases are self-recovery, and there is a role of immune system in the pathogenesis. So let's understand what are the causes of alopecia areata. So it is unknown, and the components are like genetic predisposition, stress, immune response against the hair follicle are found to be affected. Okay, and microbial, there are also evidence of involvement of microbial factors, neurotrophic factors, and adaptive and innate immunity. So to designate any autoimmune disease, any disease as an autoimmune disease, it should have demonstrate the autoimmune response to the relevant autoantigen. So that is one of the mandatory thing, and also a certain effector mechanism involving that or those autoantigens. So the having an autoantigen is an important, and your immune system like innate and adaptive immune system directed towards the specific autoantigen, that is also mandatory to determine any disease as an autoimmune. So also interesting thing is that the hair follicle is a rare immune privilege site. So immune privilege is kind of a hiding in a plain sight, like it is a mechanism of the body that protects some of the organs, vital organs, okay, from the immune system, from the involvement of the immune system. So due to absence or low expression of MHC class 1, so this the anagen the lower part of the anagen hair follicle, it remains in hiding or remains unseen to the immune system. So immune privilege blocks the immune response to the autoantigens, which are recognized by autoreactive and non-tolerized CD4, CD8 T cells. Anagen, so here, for example, we can see, so usually we know that it's all the nucleated cells in our body should have MHC class 1 expression, okay, and the antigen presenting cells will have MHC class 2 expression. So, but it is defying that rule. The hair follicle, the lower part of the hair follicle is defying that rule. And here we can see that this red marking is the MHC class 1 expression. So in the epidermis, in the upper part, we can see until the bulge, we can see there is an expression of MHC class 1 molecules. But however, in the lower part of the hair follicles, we can see there is no expression of MHC class 1 molecules. So During anagen 6 is the full grown hair fiber stage. Okay, so here we can see that there is a lower MHC class 1 or lower or no MHC class 1. There are presence of Langerhans cells, very few, and without MHC class 2. Langerhans cells are the one of the antigen presenting cells in the body, and you will find them in the lower part of the hair follicle, but they don't express MHC class 2. Also, this region, the lower hair follicle, has an immunosuppressive molecules such as TGF-beta, alpha MSH, ACTH, and IL-10, all this. And it also lacks lymphatic. So all these mechanisms are helping to generate an immune privilege site. Okay. So what happens in alopecia areata is that there is an ectopic expression of MHC class 1 molecule in the lower anagen hair follicle. So that the immune privilege which is normally present in the lower hair follicle, that is collapsed, that is lost. Okay. And it is induced after some big bang or unknown event, like nobody knows like what happens and what is the first dominant to start. Okay. So here in case of alopecia areata, we can see that there is a expression of MHC class 1 and 2 molecules all over the lower hair follicles. So in Gilhar et al, they have shown that upon the induction of with the treatment of hyphen gamma, there is an upregulation of MHC class 1. Okay. So what happens in practice is that, like this, as I mentioned before, these are the molecules which guards the immune privilege site and which allows the antigens which are antigen associated antigens, okay, they are hidden from the immune system. So when there is an increased expression of MHC class 1 molecules, so these antigens are being presented by MHC1, and there are chances that there will be interaction with the autoreactive CD8 T cells, because this autoantigen remain as in a hidden form to the immune system until now. So when they suddenly express, the immune system cannot recognize, so they starts attacking those antigens, and so in practice they start attacking those autoantigens and hair follicles, and that result in the hair loss. Okay. So in the alopecia areata patient scalp tissue, we can see in this biopsy, okay, this in the follicular, this is the follicular papilla. Okay, you can see that there is a CD4 cells are surrounding in the bulb region, and in CD8 also you can see it is very nicely populated by the inner root sheath. So we can will explain it later. Also in McKelvey et al, they've shown like in C3H HEG mice, they try to induce the interferon gamma and allow the immune activation, which result in the IP collapse and hair loss. So it suggests that activation of immune system occurs prior to IP collapse. So that is kind of not known or not very clear, like whether what happens, you know, there are the evidence of both kinds, like whether the immune system is activated first and that which leads to the collapse of the immune privilege, or whether there is an immune privilege is first collapse and which leads simultaneously the activation of immune system. So that is unclear. So based on this, the researchers have proposed like there could be a potential therapeutic strategy. What if we halt, or what if we disrupt the aberrant hair follicle antigen presentation, so that the attack which we see against the hair follicle should not happen. So for a treatment point of view, so based on that, like my previous supervisor have seen like there are the antigen presenting cells, okay, in the electron microscopy. There are close to the Langerhans cells. the antigen presenting cell, Langerhans cell, which is close to the T cells. Okay. So also they have seen that the melanocytes in the hair follicle, they have a nice interaction with the T cells. You can see that the junction is also forming at the with the T cell in the melanocytes. So it's a proposed strategy that, like, in the hair follicle epithelial cells where there is an expression of autoantigen on the MSC class 1 molecule, there will be the interaction of T cell receptor. Okay, so anyhow, if we can prevent this autoantigen coming to picture and interacting with TCR, probably we can stop the immune attack on the hair follicle in alopecia areata. So that's a strategy. So for that, it's important to understand, to have an idea like which is the key autoantigen, okay, which we can target therapeutically. So in search of autoantigens, my previous supervisor have worked a lot, more than 20 years. He worked on this topic, and he found that the antibodies in the alopecia areata, they target the trichohyalin region of the hair follicle crude extract. So this is the Western blot where they extracted the hair follicle proteins and run on the Western blot, and then they treated it with the sera of the alopecia areata patient. Okay. So here we can see there is an immune reaction against the trichohyalin region and keratin region. Also, this particular disease or like situation is also found in the horse and dog and other mammals as well. And there also they saw that the trichohyalin region was being targeted by the sera of the affected dog and horses. In 2010, they also did a advanced study based on the mass spectrometry. So what they did was they tried to collect the IgG fractions of the patients affected with alopecia areata, and they see that if the this IgGs from the alopecia areata patients are able to target the or immunoprecipitate the which protein. So they saw that there was a significant immunoprecipitation of trichohyalin. On also when they try to see the immunohistochemistry, do the immunohistochemistry, they see that the alopecia areata sera was co-localizing with the specifically with the trichohyalin region in the hair follicle. So why we think that trichohyalin could be a potential autoantigen of interest? So this is the lower part of the anagen hair follicle anatomy. So here we can see there is a dermal papilla, so which is a kind of energy house and which provides all the nutrients and growth factors to the epithelial hair follicle tissue. Okay, and these are the matrix keratinocytes. So these matrix keratinocytes are the pre-generator keratinocytes. They are not immature, and as they mature and as they grow in the, so they are pushed forward and they differentiate into the different layers of the hair follicle. So there are major 7 layers of the hair follicles. So it's such a complex, like we never might have thought, like, you know, that this is a small hair follicles and small hair fiber also have such a complex system inside. So it is, they have the 7 different layers. I won't go in much detail, but I will just try to focus on the inner root sheath, where inner root sheath is the it has around 70% of the protein in the inner root sheath is trichohyalin. So it is the one of the earliest proteins which is expressed during the growth phase of the hair follicle cycle, and it confirms the mechanical strength. The inner root sheath confirms the mechanical strength to the hair follicle inner root sheath. And also the trichohyalin is differentiating in IRS keratinocytes, which is located very close to the MHC class 1 positive connective tissue. So this is the connective tissue in blue, so which is MHC class 1 positive, and the lower part of the sorry, lower part of the anagen hair follicle is MHC class 1 negative in normal condition. So this is very close to the connective tissue. So during the maturation of trichohyalin, it goes under two different post-translation modification. Okay, sorry. So there is two key post-translation modifications involve citrullation and deamidation. So arginine is converted to citrulline and glutamine is converted to glutamic acid as the trichohyalin matures with the help of two different enzymes, PADs and transglutaminases. So how it happens is like you can see that in the immature stage, the trichohyalin, which is in red, it occurs or it is present in a globular form. So, and the keratin intermediate filaments, they're interconnected with that. So as it grows, so the structure of the trichohyalin is opened up and it slowly integrates, and it makes the it integrates makes the bonds with the keratin intermediate filament, and then it gives the mechanical toughness, okay, mechanical strength to the inner root sheath. So here we can see the lower part is an immature form, and as it grows further, we can see that the trichohyalin is giving the mechanical strength and it becomes tough, and that is only responsible for giving the shape of the hair fiber. Okay, so to see like where the exactly the trichohyalin express first in the anagen hair follicle bulb, so we did the electron microscopy. For that we take out the biopsy of the scalp tissue, okay, and then we did the electron microscopy on that. So here we can see in electron microscopic picture, so these are the trichohyalin granule, which is present in the globular form. It is expressed in this region of the hair follicle, which is very early, okay, in the differentiation stages. Also, interestingly, we saw that the trichohyalin granules, they interact with the desmosomes. So desmosomes are the proteins which gives the mechanical strength. Okay, so it's there. We saw that there is a interesting like intimate connection with the trichohyalin and the desmosomes. So let's see like what are the potential, what could be the potential citrullinated epitopes in the trichohyalin. So to check that, we what I did was like from the scalp tissue, I dissected under the stereo microscope each and every hair follicle. I dissected mechanically, and each hair follicle after separating individual hair follicles, I further dissected that into different parts like lower and upper part. Okay, so the lower part just to collect the initial trichohyalin. Okay, the early phase, which is immature, and the upper part to see the mature. And then we did the immunoprecipitation to pull out only the trichohyalin protein from the protein extract, and then we did the mass spectrometry. So the mass spectrometry results we suggested that in the lower and the upper hair follicle region, there was a different rates of citrullation. So we can see here like in the lower, okay, there is a less citrullation, and as it goes further in the upper region, we can see that there is a more citrullation sites identified. Also, we did the in silico epitope prediction using the softwares, and we identified there are totally matching with our mass spec data and in silico tools. We identified 17 potentially antigenic trichohyalin epitopes peptides, and 8 of them are native and 9 of them are native and 8 are citrullinated. So to confirm with our hypothesis that like whether the predicted peptides bind with HLA-DRB0401 molecule, we did the peptide binding assay. And peptide binding assay result shows that there are these five peptides highlighted in red have the stronger binding affinity with the HLA-DRB0401 molecule, and the four in orange, four peptides in orange have the moderate binding affinity with the HLA molecule. So then we wanted to see whether this immunogenic, whether the trichohyline epitopes are immunogenic in nature or not. So we did like the immunogenicity assessment with the HLA-DRB0401 tetramers. So this research was done in collaboration with Benaroya Research Institute in USA. So I performed the research over there as a part of the collaborative approach. So we collected the samples from HLA-DRB0401 match control, and the peptides were PBMCs were isolated and stimulated with the peptides of our interest, and they were stained with the again they were stained with the peptide HLA tetramer staining, and the flow cytometry analysis we can see if there is any positive or negative results. So here we can see that this is a pilot data I am showing, as it is unpublished data, so I am not showing all of my data here. So P1 and the peptide P1 and P1C, we can see that in control there is not much response. Okay, there is not much tetramer staining, but here in the a patient we can see there is a higher frequency of the CD4 T cell which are positive for PE positive tetramers. Further, we also did the check the ELISpot assay. In that, we collected the patients and collected their PBMCs, and the PBMC were isolated and they were treated in the ELISpot plate. So ELISpot plate, it detects the IFN gamma secretion from each and individual PBMCs. So here we can see that the control PPMCs were showing less expression of MHC class, less expression of IFN gamma, and alopecia areata patient PPMCs upon stimulation with trichohyline epitopes, they showed a higher expression of IFN gamma. So overall, summarizing my research, we found that the trichohyline is expressed during the earliest IRS cell differentiation stage in the grooving or anagen hair follicle, which is close to MHC class 1 positive cells in the connective tissue sheath. Sorry. The trichohyline granules are associated with desmosome, which suggests that like immune response or immune attack on trichohyline can also destabilize the growing hair bulb, interacting anagen growth phase and trigger hair loss. Okay, and the competitive binding assay will reveal that the 5 peptides with good binding affinity and 4 with moderate binding affinity of 17 with HLA-DRB0401 molecule. Then our preliminary function data suggests that the defined trichohyline epitopes can activate alopecia patient T cell to secrete IFN gamma, which is a pathogenic cytokine in AA. Okay. So thank you so much. I would like to thank J.A.T management, principal, and all the staff, and especially my friend Dr. Ismail. And also I would like to thank the Tobin Lab where I did all the work, Tobin Lab in Conway Institute in SPI. These are the core facilities at the UCD, and James Lab at the Benaroya Research Institute in Seattle. Thank you so much for your attention.
Thank you, sir, for a wonderful presentation that will benefit to the young researchers in your future, and lots of huge work that for hair follicles is the issue that we are facing in nowadays. So
Yeah.
thank you for sir. Thank you for the work that you have done. A very vast work and great work that you are doing here.
Thank you
Thank
so
you,
much.
sir. Thank you, sir. Uh, so any of the participant, if you have any question, any doubt, or any research information, so you can get from the sir.
Yeah, definitely. I'm open to questions.
Reggie, sir. So we have another uh, resource person. He was also delivered lecture on the genomes, plant genomes, and metagenomics. Dr. Reggie, sir.
Yes, sir.
So
Yes, sir. Yeah, it was
so
a very nice presentation, very insightful
So how are you?
presentations. Thank you, sir. Thank you, sir.
No problem. Thank you so much. How are you doing, sir?
Yeah, good, sir. Thank you.
sir is from Kerala, sir, Madras Christian
Yeah,
College,
I'm working
University
in
of Kerala.
Madras Christian College, Chennai.
Okay, okay.
Basically
Yeah,
from Kerala. Yeah.
I would like to be there, but it's not feasible to come. So
Okay.
yeah, that's why. Otherwise, I would love to be present in person and meet everybody there.
Yes.
Yes, sir. Yes, sir. We
We
are
will, yeah,
eagerly, very much, we are eagerness to meet you, sir. So we can be, when you come to India, so we will able to meet you, sir,
Sure,
and we will take guidance from you, sir, from the
sure.
research.
No problem. No problem.
Yes, sir. Yes, sir. So, any of the participants, so you have a question, so please go ahead with the, otherwise we will finish this session. And after the break, we will rejoin. So I would like to thank Dr. Shanawaz Jadeja, sir, in spite of his busy schedule, and since at a very sharp time he joined, I think so after namaz of Fajr, he had joined our session. Now the time is very like that 6 a.m. in the morning there in Ireland. है ना?
Yeah.
So, so thank you, sir, for this cooperation, and thank you for joining us.
Thank you
You
for
are
providing the
definitely,
opportunity to present my work. Thank you
yes,
so
sir.
much.
We are very much thankful to you, sir, that from you are accepted our invitation, and
It's my privilege,
and
sir.
thank you to all the participants for patience listening. Thank you, sir. Thank you, all of you.
Thank you. Thank you.
Actually, Ismail sir is busy in some meeting, so uh, we will. Reggie sir, uh, now it's time for to end the sessions because in afternoon session, see, let's see how many presenters we will get, and we will rejoin the session.
Okay, okay, sir. Even I have to go to the office. I have sent a message to you,
Uh,
sir.
Okay, okay, sir. So
I'll-
thank you for your time, sir. This
yeah,
very
sure,
much
sure.
thing
Thank
that
you,
you are
thank you.
given to me. That I am very much thanks
With
to
pleasure,
you.
sir. With pleasure, with pleasure.
Thank you, sir.
Thank you so
Thank you,
much.
sir.
Thank you.
Thank you.
Bye, bye, bye.
Bye.